Bismillahirrahmanirrahim. Assalamu alaikum friend. Today we'll discuss about genetic complementation in Pvc8 plasmid. So first of all I want to explain the structure of Pvc8 plasmid. Now this is the structure of uh, Pvc8 plasmid. So in this plasmid there are two main region are present. They are basically modified from the PBR322 plasmid. So in this plasmid, the leg Z gene present and another gene which is basically ampicillin resistance gene are also present. So what is the function of leg Z gene? So the function of leg Z gene to convert the to produce the half portion of beta galactosidase enzyme. So beta galactosidase enzyme convert the lactose into glucose and galactose. Now when we insert our desired sequence in this LexZ gene region. So what happen? This LexZ gene cannot be produced the beta galactosidase enzyme, and then the lactose are not converted into uh, glucose and galactose. So when we insert our desired gene, so this is our desired gene, and to this region, this lactose gene region. So how we can determine that our insertion are will be performed? So we use a simple test. So we take media, and this media we transfer our bacteria which are basically genetically modified, and which we insert our specific gene. So when we insert our gene, so this plasmid, then we transfer this bacteria. This plasmid are basically present in the bacteria. So when we inoculate this bacteria into our media, so in this media X-gal are present, which are the analogous of lactose. So when we inoculate this media into this this bacteria into this media, so this bacteria cannot be produce the beta galactosidase enzyme. When this enzyme are not produced, so here the X-gal, which are basically the analogous of lactose, they did not convert it into glucose and galactose, so they do not give us a blue color, so they produce a white color. So we can easily understand that our desired gene are well inserted into this plasmid. So if our recombinant DNA are not will be performed, so when we add this bacteria into this X-gal media, so they convert the X-gal into uh, glucose and galactose. So they produce blue color, and the recombinant are not be well performed. So this is all about today topic, which is basically genetic complementation and Pvc8 plasmid. So I hopefully you understand this lecture. See you in the next time.